Welcome to the discussion. And thank you for always joining us. We are going straight to our questions. This is Biology Form 1 under the topic the cell. First question, what is cell specialization? Cell specialization refers to structural modification of cells. Structural modification of a cell to perform a specific function. Question number 2. During a microscopy class, a student was unable to see the field of view. State 2 possible adjustments she needed to make to ensure that the field of view becomes visible. Number 1, she can adjust the mirror. Adjust the mirror. Number 2, ensure that the diaphragm is fully open. She can as well make sure that the objective lens, is clicked into position with the eyepiece lens. Go to question number 3. Explain how the cell membrane brings about the protective role. 1. We know the cell membrane is semi-permeable. To allow only some substances into and out of the cell. We also know that the cell membrane is charged, electrically charged, to bring about sensitivity to changes in temperature and the pH. Question number 4. Name 2 organelles that would be abundantly present in secretal cells. We have the Golgi apparatus, you can as well expect to get endoplasmic reticulum, that is both smooth and rough. You can also expect mitochondria. Remember to subscribe to this channel. Question number 5. The diameter of field of view of a microscope was found to be 6 millimeters. There were 8 cells across the diameter of field of view. Calculate the size of one cell. We know to get the size of one cell, we use the formula diameter of field of view in micrometers. You divide by the number of cells observed along the diameter of field of view. We have been given the diameter of field of view to be 6 millimeters. So we take 6 millimeters, we multiply with 1000 to change them into micrometers. Then we divide by the number of cells observed, and we have been told we observed 8 cells. This gives us 6000 divided by 8, giving us exactly 750 micrometers. That is, we got to be state 2 differences between electron microscope and the light microscope. For the purpose of showing the differences, we can use a table. One side we have the electron microscope. Another side we have the light microscope. We know the electron microscope has high resolving power. A light microscope has low resolving power. We know an electron microscope uses a beam of electrons. To illuminate the specimen. A light microscope uses light to illuminate the specimen. We move to another point. This one uses electromagnetic power. A light microscope uses a glass lens. The last point we can say is that this one has high magnification power. This one has what we call lower magnification power. Question number 6, state the functions of the following organelles. We have the vacuole. The vacuole stores various substances, including waste materials, or what we can call products. Number 2, it contributes to osmotic properties of the cell. We have Roman 2, the cell membrane. 1, controls exchange of materials between the cell and its environment. Number 2, it also forms the outer boundary. In an animal cell. We go to question number 7. Name the organelle that performs the following functions: digestion and destruction of worn out cell organelles. These are lysosomes. We go to B, osmoregulation. This is the contractile vacuoles. Question number 8, explain why the following processes are important during preparation of temporary slides. Number 1, we have staining. Staining is done to make cells more visible. Use a sharp cutting blade. This is to prevent distortion or destruction of cells. Cutting very thin sections. This is to allow light to pass through. For easy viewing. We go to question number 9. In an experiment to establish the size of an onion cell, a learner observed the following on the microscope field of view. If the student counted 20 cells across the diameter field of view, calculate the size of one cell. We know that the size of the cell is given by diameter of field of view in micrometers. You divide by the number of cells. In this case, when you are not given the diameter of field of view, then you count the number of spaces given. A single space is equivalent to 1 millimeter. This is also another millimeter, another millimeter, another millimeter. That means this gives us 4 millimeters. We multiply by 1000 to change them into micrometers. To divide by the number of cells counted. This one will give us 4000 divided by 20, giving us exactly 200 micrometers. Question number 10. 10 part A. You differentiate between magnification and resolution of a microscope. Magnification is simply the ability of a microscope to enlarge what we call tiny specimens. To enlarge tiny specimens while resolution power is the ability of a microscope to separate between two tiny structures to appear distinct. We also have another question here. Differentiate between mounting and staining. Mounting, by definition, mounting is actually the placing of a prepared slide onto the stage of a microscope, whereas staining refers to use of a chemical stain on the specimen for a clearer observation. Thank you for watching and welcome to this discussion. And in case you are watching this channel for the first time, please take a second or two and click the subscribe button so that anytime we publish a video like this, you will automatically be given a notification.